Welcome everyone. This is Dr. Shruti Analyx, Assistant Professor, Department of Biomedical Engineering, SRMIST, Katangattur. So today we'll be looking at the topic regeneration of cornea and injury-induced regeneration in cornea and regulation by epithelial stromal interaction. So these are the topics that are covered in the Unit 4 of ectoderm-derived organs. So first, let us look at the corneal epithelial function. So the corneal epithelium, which is found in the eye, it is the outer protective layer, and it helps to make sure that there's no dust particles or any other factors that are injuring the inner part of your eye. This also helps to maintain the transparency. So there is rapid turnover that is seen in the corneal epithelium, and this is due to the exposure to environmental stresses. So we see that our eyes are constantly exposed to a lot of environmental factors like dust molecules or things that can poke your eye. So to protect it, and to make sure that the normal count of the cells are always maintained, so the cells need to maintain a rapid turnover. So to help in the corneal epithelial regeneration, we have something called the limbal stem cells. The limbal stem cells reside in the limbal palisades of what, which is a specialized niche at the corneoscleral junction. So this is the cornea of the eye. This is the sclera region. At the junction, we have the limbal stem cell. So at the junction of the sclera and the cornea, we have the limbus, which will have the limbal stem cells. So the periphery of the central region is where your limbal stem cells are residing. Reside in the limbal palisades of Vogt, which is a specialized niche at the corneoscleral junction. They have the ability to self-renew to sustain the corneal epithelium and to prevent the conjunctival overgrowth. Why do we need to maintain or prevent the conjunctival overgrowth? Conjunctiva is the mucous membrane which is covering the white of the eye. So the white of the eye is not transparent, right? So this naturally grows towards the cornea. But what the limbal stem cells do is that they act as a barrier and ensure that only the corneal epithelial cells are covering the corneal. So it is preventing the conjunctival cells from overgrowing towards the transparent corneal region. So what happens if there is a failure in the limbal stem cells? So in those cases, the conjunctival cells will invade the cornea, and this will cause the conjunctivalization, and this results in the vascularization, abnormal blood vessel growth, scarring and opacity of the cornea. So thereby there is hazy non-transparent cornea and also loss of vision. So the cornea must stay clear for the light to pass through. So the LSEs help to maintain corneal clarity by blocking the conjunctival tissue, and limbal stem cell deficiency can lead to blindness if it is untreated. Okay, so it is very important to prevent the conjunctival overgrowth. So the limbal stem cell also help to repair the damage that is there in the cornea by migrating centripetally to replenish the corneal epithelial cells. So what are the different types of corneal injuries that can take place? So there can be mechanical injuries, like there can be a scratch, abrasion, lacerations, etc. Or there can be chemicals that are accidentally introduced, or it basically falls on your eye. So acid or alkali damages can be there, and there can also be infections, infectious keratitis. So bacterial, fungal, viral infections can also affect the cornea. The consequences of the limbal stem cell deficiency. So when there is a limbal stem cell deficiency, what happens is there is impaired corneal healing, and this results in the persistent epithelial defects, which is a non-healing wound. And there can also be conjunctivalization, which is the abnormal blood vessel and conjunctival cell invasion, and corneal opacity and scarring, which results in the vision loss. We just discussed that. And what are the clinical importance of the limbal stem cells? So we can use autologous limbal stem cell transplantation. The same donor, we can basically extract the limbal stem cells. We can expand the population of the stem cell and transplant it back into the person where the defect is experienced. So this can restore the vision in the case of limbal stem cell deficiency. So to obtain the autologous stem cells, there can be different methods that can be done. So either the person's own stem cells that were frozen from the frozen stem cell bank that was frozen at the birth of the person, or this can even be, let's say, in certain cases when the stem cells are not sufficient in amount, or are not of good quality, the adult stem cells can be taken and then they can be reprogrammed to become the specific stem cell type that is needed. So, ex vivo expansion of the limbal stem cells can be done using amniotic membrane or scaffolds. So, amniotic membrane is used because of the ability to support stem cell characteristics, because it is taken from the newborn. Proliferative capacity that is maintained by this type of a scaffold is very high. And same way, we can also use artificial scaffolds using certain natural polymers along with certain growth factors to maintain the good limbal stem cell population. So ex vivo expansion is also used to help in regenerating the limbal stem cells. The limbal epithelial stem cells, which are present at the periphery of the cornea, palisades are worked, and as they differentiate, they are moving towards the center of the eye. The limb stem cells, they migrate to the central cornea. So first they are forming the transit amplifying cells, and as they move towards the center, they are forming post mitotic cells, and even as they move further, they form the corneal epithelial differentiated cells. And we can here also we can see that the transparency is maintained. I had discussed why the transparency appears in these type of cells. Transparency in the lens cells and the corneal cells are due to the absence of any organelles. So as they are moving towards the center, they start to lose their organelles like nuclei, mitochondria, and endoplasmic reticulum, so as to reduce the scattering of light. They also have certain specific proteins that are helping in the uniform refractive index, and they also have ordered packing. So the ordered packing makes sure that the refractive interfaces are reduced, and thereby the bending or scattering of the light is also reduced. So next, coming to the corneal regeneration injury induced models, the epithelial cells will migrate and proliferate, and then there is stromal remodeling followed by the collagen deposition that takes place. There's also a major role that is played by the immune cells such as macrophages and neutrophils that are helping in the wound repair. So we will see them one by one. So first, coming to the epithelial wound healing. So after the injury takes place, the corneal epithelial cells will migrate from the wound edges. So your epithelial cells are found towards the periphery, and they will migrate from the wound edges to cover the defect. Then the proliferation of the basal epithelial cells will help to restore the epithelial layer. So the basal layers will proliferate, move to make up for the cell loss. So proliferation of the basal epithelial cell restores the epithelial layer. When the old cells are lost from the surface of the cornea, this is being replenished by the basal cells, which are moving centripetally, and they are helping to recover the or regenerate the corneal cells. So next, coming to the stromal response, the stromal keratocytes transform into fibroblast and myofibroblast, which help to aid in the repair. So these cells secrete extracellular matrix components such as collagen to restore the stromal integrity. And then there is also nerve regeneration which is needed for the cornea. So the corneal nerves regenerate to restore the sensitivity and to support the epithelial health of the cornea. So to help in this regeneration process, we can use biomaterials as well as scaffolds. Some of the biomaterials that can be used are hydrogen based carriers for cell transplantation, and because of the ability to mimic the natural ECM, so these hydrogels have been very much useful, and they can also incorporate growth factors and other cell signaling molecules, and they also help in the easy movement of molecules in and out of the cells. The next one is a decellularized matrix. This is a matrix or an ECM wherein the cells have been removed. Decellularized matrix are formed by extracting tissues from healthy donors, or it can be even after the death the tissues are taken, and from the tissue the extracellular matrix alone is retained, whereas the cells are removed. And then this decellularized matrix can be used to implant cells from the uh, patient into which this cells have to be transplanted. So basically you can this can be reduce the possibility of immune rejection, because the matrix that you're using is taken from the original tissue. So they are the complexity of the extracellular matrix is maintained, and thereby it is able to support the cell growth and proliferation to a much better extent. So they can be used to support the epithelial growth. So next, coming to the epithelial stromal interaction. So the epithelial stromal interaction refers to the bidirectional biochemical and mechanical crosstalk which exists between the epithelial cells that are lining the tissues and organs and the surrounding stromal compartment. So which is the stromal compartment is basically the supporting connective tissue. This dynamic signaling between the epithelial and stromal cells will help to regulate different phenomena such as the organ development, tissue homeostasis in adults, and also in the wound healing process. And for it also plays a major role in several disease progression. So how does this interaction take place? So the epithelium will release growth factors like EGF, uh, FGF, and these will help to activate the stromal fibroblasts, and the stroma in turn will release certain factors. So the stroma will modulate the repair of the epithelium via the release of certain cytokines like TGF beta, interleukin 1, and it also helps in the extracellular matrix remodeling. In most cases, the injured epithelial cells will release the EGF and FGF, activating the quiescent stromal fibroblast. These growth factors in turn will trigger the fibroblast proliferation, migration, and production of the provisional ECM such as fibronectin and collagen, and thereby it will create a scaffold that is needed for the repair. So in turn, the activated fibroblasts that are found in the stroma will secrete the TGF beta and interleukin 1, which will signal back to the epithelial cells. The TGF beta promotes keratinocyte migration and wound contraction, while the interleukin 1 helps to amplify the inflammation and recruit the immune cells. So, concurrently, the fibroblasts remodel ECM via the matrix metalloprotease, and this will release the bound growth factors that sustain the epithelial repair. So this is how the epithelial stromal interactions are working. So what are the key regulator molecules that we are discussing about? The first one is the transforming growth factor beta, TGF beta. So this is a dual edged molecule, as in it is essential for wound closure, that is the myofibroblast differentiation, collagen deposition, etc. But an overactivity of this TGF beta can drive the pathological scarring, that is hypertrophic scarring, or it can also lead to fibrosis of the cornea. Next, coming to the FGF, EGF signals. So these are the primary drivers of the epithelial proliferation and migration, and also in the reepithelialization process. Stromal fibroblasts amplify these signals through paracrine loops, which are help in accelerating the healing process. So the balance is very critical. So when there is a dominance of the TGF beta signal, so there is excessive ECM deposition, scarring, and tissue contracture can take place. Whereas a low FGF EGF signal can lead to impaired epithelial closure, chronic wounds, and also infection risk is very high. So we end this session on the note that a harmonious crosstalk between these growth factors help to ensure rapid scarless repair, and if there is any dysregulation, this tips the healing towards fibrosis or failure. So that is why in general, when there is a scarring that happens in the adult, this usually tends to lead to fibrosis or failure. Thank you for listening.